In vivo interaction between flag-tagged protein P and chaperone H was examined by co-immunoprecipitation. Co-chaperone A was included and co-IP was performed in the presence or absence of cyclohexamide. The result of co-IP experiment are shown below, and from this result we need to determine their interaction. From this we can understand that protein P is tagged with the flag. Looking at the first picture, in the first lane, chaperone H and co-chaperone A are present. The same condition is seen in the third picture. In the second lane, flag-tagged protein P and co-chaperone A are present. In the third lane, flag-tagged protein P and chaperone H are present. In the fourth lane, flag-tagged protein P, chaperone H, co-chaperone A are present. In the last lane, all the components are present. Do you know what flag is? It is a short peptide tag that is artificially attached to protein P. Protein P itself may not have a good antibody, so researchers add a flag tag to it. This tag does not change the function of protein P but allows us to specifically detect or isolate it using an anti-flag antibody. So flag tagged protein P means that protein P is fused with the flag tag and can therefore be specifically recognized by an anti-flag antibody. Then what does IP flag means? IP flag refers to immunoprecipitation using an anti-flag antibody. The anti-flag antibody binds only to flag tag. Therefore, it pulls down only the flag tagged protein P. Any other protein or any band visible for another protein in the IP lane must be present because it is physically associated with the protein P. The second picture represents immunoprecipitation using anti-flag antibodies. Thus, flag-tagged protein P is precipitated, and if any other proteins interact with the protein P, they will also be precipitated along with it. In the first lane, there is no band because flag-tagged protein is absent, as shown in the first picture. Since flag-tagged protein P is not present, no band appears in this lane. In the second lane, flag-tagged protein P is present, so its band is visible. No other bands are seen. Although co-chaperone A is present in this lane, its band is not visible. This is because immunoprecipitation is performed using anti-flag antibody, and only protein that interact with the protein P will be detected. Since co-chaperone A does not interact with the protein P, no band appear for it. If it were interacting, a band would be visible. In the third lane, protein P and chaperone H are present, and both the bands are visible. This indicates that chaperone H interact with the protein P. In the fourth lane, protein P, chaperone H and co-chaperone A are present. Here the band for a co-chaperone is also visible along with the protein P and chaperone H. Since we already established that co-chaperone A does not directly interact with the protein P. The appearance of this band for co-chaperone A suggests that co-chaperone A interact with the protein P in the presence of chaperone H. Based on this observation, the first option is correct because protein P interact with the co-chaperone A and chaperone H. The second option is that chaperone H interacts only with the newly synthesized protein P. To find whether this is correct or not, we must understand the role of cyclohexamide. Cyclohexamide inhibits protein synthesis. That means, with the cyclohexamide, the synthesis of new proteins is blocked. So, any detected interaction would involve only pre-existing proteins. In the absence of cyclohexamide, both the newly synthesized and pre-existing proteins are present. Chaperone H is present in the lane 1, 3, 4 and 5. In the first lane, there is no protein P, so band for the chaperone H is not visible. In the third and fourth lane, the band is visible, but there is no band in the last lane. From this, we can understand that the band for chaperone H is visible only in the absence of cyclohexamide, and if cyclohexamide is present, no band is visible for it. This suggests that in the lane 3 and 4, cyclohexamide is not present to inhibit the protein synthesis and therefore the band is visible. In the fifth lane, cyclohexamide is present and it inhibits protein synthesis, so no new protein P is formed and therefore no band is visible. The third option, which states that interaction between co-chaperone A and protein P is independent of chaperone H, is incorrect. Co-chaperone A can associate with the protein P only if chaperone H is present. The fourth option is correct because the interaction between chaperone H and protein P does not require co-chaperone A. This is evident from the lane 3 where the band for chaperone H is visible even in the absence of co-chaperone A.